So now I would call on our next speaker, who is Eyðfinn Magnussen, who is Associate Professor at the University of Faroe Islands, working in the field of fish ecology, with a special research interest in life history of fish, biodiversity, population genetics, zoogeography, and temporal and spatial distributions. And Eyðfinn is going to be talking about the Faroe Bank as a field study for cod in relation to climate change. So the floor is yours, Eyðfinn.
Yeah. First of all, I wish to welcome you to my lecture, which I have called Faroe Bank and Oceanic Field Laboratory, where I'm going to present some results I have found on the bank during the last year I have been working with the area. My presentation will be in 4 main subjects. I start with a general description of the Faroe Bank area, and then I give a short presentation of some of the special characteristics which have been involved for some of the fishes living in the area, and continue in genetic analysis. And at the end, I will give a presentation of the fish assemblage on the bank and tell something about the changes that occur in the species distribution in the area during the last decades. Let's start with description of the Faroe Bank. This picture shows the bathymetric condition in the North Atlantic. In the middle, we have the Faroe Island, and here we have the Faroe Bank. Faroe Bank are located southwest of the Faroe and is isolated from the Faroe Plateau by the Faroe Bank Channel, which Boy Hansson also was talking about yesterday. The water masses on the bank are flowing around on the bank, which make the Faroe Bank as its own ecosystem. Because of this isolation, fishes on the bank have in many cases evolved special characteristics which make them different from those in the other area. The most famous of this characteristic is probably the high growth for the cod on the bank. This picture shows cod from the Faroe and in different areas around the Faroe Island. At the top, we have the Faroe Bank cod, which, when they are 3 years old, have an average size on 4.2 kilogram. Here, we have the cod from the Faroe Plateau, which have a weight on only 1.7 kilogram. And at the bottom, we have the cod from the West Greenland. So as you can see, there are a huge difference between the size, the growth of this cod in different areas. But the high growth found for the cod seems not to only to be the case for the cod, but seems to be a general phenomenon for most of the stationary fish living on the bank. This figure is from a paper which I was published 1 year ago, where I compare growth of Faroe Bank fishes with the growth in those species in other areas. And in general, it's shown that fish species which are stationary for the Faroe Bank in average growth 36% higher, faster than those in other areas. There were 3 main reasons for the high growth for the fishes, Faroe Bank fishes. One of them was that the temperatures are relatively high on the bank. Additionally, the fish seems to have a lot of food. And the last one was that the fish also seem to have very good growth characteristics. So let's continue with some genetic analysis, which show that the fishes on bank are genetically different from those found in other areas. This picture is from An investigation made by Einar Magnussen from the Danish Fishery Research. Einar collects samples from different cod stock in the East Atlantic. He also includes cod from the Faroe Bank as well on the Faroe Plateau. When he compared the different genetic results, he found that the Faroese cod was completely different from those found in the other area. Additionally, he also showed that there were differences between Faroe Plateau and Faroe Bank cod. If we continue to another species, which is called poor cod, this figure shows results from genetic analysis made on this species from different places in the North Atlantic. And as for the cod, it also shown that the Faroe Bank poor cod is completely different from those found in other area. The next topic I will talk about is the pen gene, which we worked on for a couple of years ago. And the results were published in a Canadian journal in November last year. The pen gene is one of the most used biomarkers for discriminating between different cod species in the North Atlantic. And there are found two different types of this pen gene. We called it pen A and pen B. The question which was addressed in our paper was, how good is the pen gene as a biomarker? Here we show the allele frequencies, distribution of allele frequencies in the North Atlantic. And it's shown that there was very low frequency in the north and very high frequency in the south. In this area, there was a huge difference in the temperature condition. And when the frequencies are plotted against temperature, there seems to be a very high correlation between with the temperature, which shows that the allele probably are under selection pressure, which are not so good for biomarker. So we try to investigate if this allele frequencies were driven with the temperature. If that was the case, they are not so good for biomarker. The first thing we was investigate how this allele frequencies was affected by the temperature. Here we see the temperature condition around the Faroe during the last century. And you can see, as you can see, there will be a huge variation. In 1907, the average temperature in June was on 9.5 degrees, while it 25 years later had decreased by 2 degrees. And if the pen gene are affected by the temperature, we would expect that this would have effect, in some way had effect the allele frequencies. So we collect fresh sample from blood and muscles, as well as historical data. The historical data was taken from otoliths. Otoliths are normally used for age reading, but they also contain DNA, which could be used for DNA analysis. On the fishery laboratory, there's a lot of these otoliths. So using those, it's possible to go back in time and see how the allele frequencies have been changing during the time.